Hi everybody, welcome to part 2 of the week 4 video lectures. This is on mitosis and the cell cycle. So now we're talking only about cell division in eukaryotic cells, and this is specifically the type of cell division that occurs in your body cells or somatic cells for growth and repair. So remember that the whole idea behind mitosis is to create two identical daughter cells from one parent cell. And so we are going to talk about a little bit about how that process happens. We're going to talk about the cell cycle, and the cell cycle is everything that happens between one cell division and the next cell division, and that's divided into different phases called interphase, mitosis, which we'll concentrate mostly on, and cytokinesis. So cytokinesis is the actual division of the cells. Cyto, C-Y-T-O, means cell. So anytime you're reading about science, especially biology, if you see cyto, think it must have something to do with cells. And kinesis is the division. We'll talk a little bit about the stages of mitosis and the regulation of the cell cycle as we go through all of this. But of course it means that we have to talk about some complicated topics, and I wanted to remind you of a few terms that we've briefly mentioned but you want to have stuck in your head. So we're going to talk about haploid and diploid numbers of chromosomes. So haploid means there's one copy of every chromosome. And in fact, there are some organisms that have a major part of their life cycle where they're haploid. Humans happen to not have a long phase of haploid. We are diploid organisms, and that means we have two copies of every chromosome. And that is because we got one copy of each chromosome from each of our parents. So we have two copies of every chromosome. Now, when we talk about haploid, we usually use the term N, and for diploid, 2N. And the reason for this is because every organism has a different normal number of chromosomes. So on this slide, I'm showing you a few different organisms. In the upper right, you see humans. And humans have a haploid number, or N, is 23, and our diploid number, 2N, is 46. Just to point out that this is not a magical number, fruit flies have 4 chromosomes, so their diploid number is 8. Peas, thank you to Gregor Mendel for me choosing peas, have 7 chromosomes, so their diploid number is 14. And I went to try and find a large number of chromosomes in an organism that everybody would know about, and I figured we probably all know what a turkey is. Turkeys have 40 chromosomes, so their diploid number is 80. Okay, so the number, it doesn't have anything to do with how an organism works. It's just the way that their genes are organized on chromosomes. And so in order to compare the genome of, say, a turkey to the genome of a human, we don't want to be thinking about the total number of chromosomes, but whether or not we're talking about a haploid, N number of chromosomes, or diploid, 2N number of chromosomes. All right, so now that we've got that terminology, let's talk a little bit about what happens during the cell cycle, and we'll be talking about cell cycle in humans here, but in general, any organism that's eukaryotic is going to have a cell cycle, and that cell cycle is everything that happens between one time the cell divides and the next time it divides. All right, and so the largest part of the cell cycle is generally the interphase portion. So interphase is this region here where it's kind of this greenish color, and interphase is between from the end of cell division, cytokinesis, to the beginning of mitosis. So it's everything that's happening in between cell divisions, interphase. Now, a lot of interesting things happen during interphase. For the entire period of interphase, the cell is doing all of its normal metabolizing. It's using nutrients, it's creating wastes, it's making proteins, it's doing all the things that cells do. But interphase is divided into 3 sections. So there's G1, S, and G2. And these terms go back to when early microscopists were looking at cells, and they noticed that during mitosis and cytokinesis, all kinds of exciting things were happening with the chromosomes. They were moving around, dancing around, doing all kinds of crazy things. But in between, it was kind of boring. But later scientists realized that during the middle of interphase, there was a period of time when the DNA was synthesized. It was copied because they could detect that the amount of DNA inside the cell doubled. Okay, and so the period of time when cells are copying their DNA is called S, and S stands for DNA synthesis. And S phase is in interphase, okay? And so the regions before S and after S were called G1 and G2 for gap 1 and gap 2, because certainly when you're looking through a microscope, it didn't seem like anything was happening. So there was a gap in interesting activity as far as microscopists were concerned. Don't make the mistake of thinking that means nothing is happening. During G1, the cell has to grow, make lots of proteins and lipids and carbohydrates, and get ready to divide again. So if you think about it, you have a big cell and it divides in half. Each of those 2 cells is now half the size of a normal cell. So they are going to have to grow. And so until the cell finishes growing back to the size that it was before division, it's not going to start copying its DNA. After DNA synthesis during G2, the cell is still making proteins, it's still metabolizing, but it starts to make some new proteins and getting ready to divide by mitosis, or if it's going to go through meiosis, by meiosis. So there are things that are happening there. I did want to point out there's this little region here where there's an arrow coming off of the G1 part of interphase that's called G0. So sometimes cells do not prepare to divide again. And so they end up in this phase that's called G0, where they're metabolizing, they get all kinds of things happening, but they're not preparing to divide. Now they could get a signal at some point that will have them leave G0 and go back into G1 and get ready to go through interphase, or through S phase and mitosis again. And we'll talk about that a bit more when we talk about cell communication, which is going to be several weeks from now. Okay, so interphase has a lot of things happening, and then of course interphase is followed by mitosis, where the chromosomes divide, and cytokinesis, where the rest of the cell divides. Now, if we're going to talk about dividing the chromosomes, we have to define what we mean by chromosomes. And so in this slide, I wanted to point out that a chromosome, and here's a copy of a picture of a chromosome up at the top in red. A chromosome is a DNA double helix that's working, it's been wrapped around some proteins, and it is a single long DNA molecule. Now, during S phase, in interphase, this DNA is copied, and you end up with a chromosome. So this is a chromosome, and this is a chromosome after it's been duplicated, where there is the original red DNA double helix, and the new one is, in this case, in this kind of pinkish color. This is one chromosome made up of 2 sister chromatids. And after mitosis, these 2 sister chromatids will be separated, and they will each become a chromosome. All right. Now, in this figure, they're showing the 2 chromosomes next to each other, but remember that each chromosome, the red chromosome is going to be in one daughter cell, the pink chromosome is going to be in the other daughter cell. They're not going to be right next to each other anymore. Now, we also have to talk about what a homologous chromosome is, because this is going to be important, especially when we start talking about meiosis in the 3rd video. So homologous chromosomes are shown here in green and in purple. And they're the same chromosome. They have the same sequence of genes on them. It's just that one homologous chromosome came from each parent. All right. So again, after the DNA is copied, which the process is actually replication, we'll talk about that there. So that's why it says duplication and replication. They both mean we copy the DNA. You will end up with 2 chromosomes that are homologous. So we've got the green homologous chromosome made up of 2 sister chromatids and the purple chromosome made up of 2 sister chromatids. Okay. The other thing I want to point out that will be important to keep in mind is that the X and the Y chromosomes are homologs. Now, if you've ever seen a picture of an X and a Y chromosome next to each other, you will know that the X chromosome is much bigger than the Y chromosome, but they do have some sequences near their centromere, and the centromere is this region in the middle, all right, of the chromosomes. So I kind of think of it looks like the waist when you look at a picture. Here they're drawn as circles or discs, but there are a lot of proteins that are holding these sister chromatids together in the center. They're also actually held together along the arms, but it's just not shown in these cartoons. But the regions near the centromeres of the X and Y chromosome that are homologous, and so X and Y chromosomes are truly homologous chromosomes. Okay, so we've defined a bunch of terms. Let's move on and talk a little bit about the stages of mitosis. And I'm going to go over this relatively quickly. I have posted some videos in the resources section of week 4's overview page, and you can get more details there. I've divided them up into some videos by the Amoeba Sisters, which are kind of cartoony and funny, and Mr. Anderson, which are more serious. But both series of videos have a lot of good information and they're accurate, so good to look at. And then, of course, don't forget to read your textbook. So keep the term PMAT right down here in mind. Prophase, metaphase, anaphase, telophase. That's the order of the stages. Some people put another phase in between prophase and metaphase that they call prometaphase, because it's halfway between prophase and metaphase on a picture like this. And the other thing I want to keep in the front of your minds is that these aren't actually discrete steps along the way. This process is a continuous process, but we as humans recognize certain points in this process where things are significantly different, that they are given a name. So for instance, in prophase, the first phase here, the chromosomes that have been duplicated already in interphase are starting to condense. They're getting shorter and thicker, so we start to be able to see them as individual structures. The nuclear envelope around the DNA starts to break down, and the mitotic spindle starts to form. So we're starting to get these microtubules. These yellow structures are microtubules that are starting to form. Right now they're still outside of the DNA, but they're starting to form. When all of the chromosomes are condensed as far as they are going to become condensed and are attached to mitotic spindle fibers, one fiber is attached from each pole of the mitotic spindle. There is this pushing and pulling of the chromosomes that get them all lined up in the center of the cell on this imaginary line. So this dotted line here is an imaginary line that's called the metaphase plate. When they're all lined up like that, the cell receives a signal that tells it it's time to divide the chromosomes. And so the chromosomes divide in and get pulled to each end of the cell by the mitotic spindle fibers. And after anaphase, we have the two new nuclei starting to form. You can see the 2 new nuclei here starting to form. And there is a cleavage furrow. That's this indentation between the cells so that the cell starts to divide. Basically, cytokinesis begins as telophase is ending, so they are frequently shown together. But basically, cytokinesis is the cell just starts to pinch. Okay, it's as simple as that. I will post another video by Drew Barry, the guy who did that apoptosis video, in the resources about what it looks like as this process is happening. So look for that. It is really cool. Meanwhile, I want to move on to cell cycle regulation. And what these are steps in the cell cycle where the cell stops, senses whether or not it's ready to move on to the next step. So we've already talked about this M checkpoint right here in the middle of mitosis. That is what happens at metaphase, when the chromosomes are all lined up in the middle, and it's not until they're all lined up in the middle that anaphase starts to separate the sister chromatids into individual chromosomes again. Okay, if they're not evenly lined up, the cell does not want to start dividing the chromosomes because it wants to make absolutely sure that each of the daughter chromosomes is going to get the right complement of DNA. There's also a checkpoint in G1. So here's the G1 checkpoint. And the G1 checkpoint is a place where the cell takes note and says, have I grown to the right size? Do I have the right proteins present? Does my DNA look like it's in good condition? Because it does not want to replicate its DNA if the DNA is not going to be good for the daughter cells. Cells that do not pass the G1 checkpoint are going to enter that G0 phase that I mentioned, where they're metabolizing and living, but they're not getting ready to divide. If the cell does pass that G1 checkpoint, it will enter S phase. It will copy its DNA. When it finishes that, it'll enter G2 phase. It will go through all the things that happen during G2. And at the end of G2, there's another checkpoint, another important checkpoint right here called the G2 checkpoint, where the cell stops and says, am I actually ready to start mitosis? And only when everything is ready and the cell can pass that G2 checkpoint will prophase start. So this has been an overview of mitosis and the cell cycle, and in the next video we will talk about meiosis.